Welcome to the Chemistry of Life with Lipids. My name is Karam,
I'm Ross.
and also in our group we got Hirsh, Jane, and Safa. Well, first we have to begin with what exactly is a lipid, Ross?
Well, a lipid is a term to describe a range of biological molecules.
Well, what are some characteristics of the lipid?
Well, they're hydrophobic, or insoluble in water. They are soluble in nonpolar solvents. They contain carbon, hydrogen, and oxygen.
Well, I heard that there are three types. Could our guest star please tell us something about those?
Well, yes, there are three types: triglycerides, phospholipids, and lastly steroids.
Tell me a little bit more about triglycerides.
Triglycerides are composed of three fatty acids and a glycerol molecule. Each of these three fatty acids takes the place of one of the three OH groups on the glycerol. In the scientific community, glycerol is also known as 1,2,3-propane triol. The fatty acids are simply long carboxylic acid chains.
And if you guys remember from the lectures in class, this is directly related to the process of esterification. It's important to note that the three fatty acids are not all the same. They're different because they are designated R1, R2, and R3. These all can differ in two ways, which give them their individual properties. The first being the length of the hydrocarbon chain and the number and position of the carbon-carbon double bond, which differs between saturated and unsaturated fats. Now that we learned about triglycerides, tell me a little bit about phospholipids.
Well, phospholipids are exactly like triglycerides, except instead of having three fatty acid chains, there's only two fatty acid chains and a phosphate molecule that takes the place of the third one.
Also, different phospholipids vary in their fatty acid chain lengths and in the way that their molecules are attached to the phosphate. This is how we differentiate between the different ones. Yo, Karam, sometimes I hear my parents talking about how there's like a good cholesterol and a bad cholesterol. Can you explain the difference between HDL and LDL cholesterol to me?
Well, to start off, HDL stands for high density lipoprotein, while the LDL cholesterol stands for low density lipoprotein. The LDL is what is often labeled as the bad cholesterol because it's known to be responsible for depositing cholesterol inside of the arteries, which increases the risk of heart disease. Also, it's produced by eating saturated fats as well as trans fats. The HDL cholesterol, on the other hand, is labeled as a good cholesterol because of its responsibility of removing the cholesterol from the blood and returning it to the liver. It is produced by eating monounsaturated and polyunsaturated fats. Ross could tell you a little bit more about the saturated fatty acids and their structure.
Well, as you see in the image below, the saturated fatty acids are made of a carbon chain. All the carbon-carbon bonds are single, so the bond angle is always tetrahedral. Because of the structure, they can easily pack together. This results in higher van der Waals forces, which leads them to have high melting points and turn solid at room temperature. Examples of saturated fatty acids are butter and lard.
Unsaturated fats, on the other hand, have carbon-carbon double bonds, which is what creates the kinks and the lack of stacking that can take place in the molecule. As a result, these molecules have less van der Waals forces and have lower melting points, turning liquid at room temperature. Some examples of this include corn oils and oils found in plants and fish. Below is an image of an unsaturated fat, and you can see the double bond in the carbon towards the right. This is an image of a linolenic acid above and a linoleic acid below. You can notice the position of the double bond, with the linolenic acid having it at the third carbon, while the linoleic acid has it at the sixth carbon. This is how you come up with the name omega-3 or omega-6 fatty acid.
Both of these fatty acids cannot be made by the body, therefore they must be acquired by dietary means. They are involved in metabolic processes that make prostaglandins in order to reduce blood pressure. These are unsaturated fatty acids promoted by the production of HDL, which can lower cholesterol levels and reduce the risk of heart disease.
These two hydrocarbon chains serve as hydrophobic tails, meaning that they are water repellent. The rest of the phospholipid is then hydrophilic, which means that it wants water and is attracted to it. Then, these can form a phospholipid bilayer. These are frequent in the human body in the formation of the membranes of cells, the hydrophobic ends staying away from the water, while the hydrophilic ends maintains contact with the cytoplasm of matrix between the cells. As you can see in the image, that is an image of the phospholipid bilayer with the hydrophobic and hydrophilic ends.
Moving to iodine number, we can define it as the number of grams of iodine which reacts with 100 grams of fat.
This essentially serves as a measurement of how unsaturated a molecule is.
These unsaturated fatty acids can undergo addition reactions by breaking the double bond and adding incoming groups.
On this slide, you could see an example of an addition reaction, with ethene reacting with bromine.
This essentially is the same thing that we do with iodine number, except instead of bromine, we have iodine.
Moving on to the next type of lipid, a steroid.
Steroids are made of bonds, not like berry bonds.
Steroids contain 4 hydrocarbon rings. They include a long hydrocarbon chain, 4 rings, and a hydroxyl group. Cholesterol is very important in the body as it is used in the synthesis of other steroids, including sex hormones. Ooh. These also alter the permeability of the membrane.
As was described before, in order to form a triglyceride, there's the condensation of glycerol and 3 fatty acids involved. Basically, the glycerol, which has 3 OH groups off of each of its carbon, then has those OH groups eliminated, and those turn into water. As a result, esterification occurs, and this turns into the 3 fatty acids link to the 3 places where the OH was originally. The OHs break off, and 3 water molecules are formed.
Fats and oils are insoluble molecules, so they are broken down to their component molecules, which are fatty acids and glycerol, in the process of digestion.
This involves hydrolysis reactions in the presence of water, under the control of enzymes known as lipases.
Lipases are secreted in different parts of the gut, which act sequentially to complete the digestion of the lipid as it passes through. The slowest molecules to be broken down may take many hours before they are made soluble and can be absorbed into blood.
These are enzymes that are made of protein and are very sensitive to changes in pH of the medium. Controlling pH in the gut is one way in which the body controls lipid digestion.
Lipids contain stored energy, and this can be released when they are broken down in the reactions of respiration. Because they're less oxidized than carbohydrates are, they can undergo more oxidation. When they do that, they can release more energy per unit mass when they're used as the respiratory substrate. The significant difference here is that there's almost twice as much energy released by lipids as is released by carbohydrates. However, lipids are also insoluble, and so this energy is not as readily available as the energy in carbohydrates. There are more reactions involved in their breakdown. Specifically, as we learned in health, lipids account for approximately 9 calories per gram, while carbohydrates account for 4 calories per gram.
Lipids have several uses in the body, including energy storage, insulation and protection of certain organs, steroid hormones, structural component of cell membrane, and also certain lipids can help reduce the risk of heart disease, lowering the levels of bad cholesterol, which was LDL, and can alleviate other diseases.
Some hormones, such as sex hormones, are made from the lipids in the form of steroids. Also, bioacids, which aid digestion in the fat in the intestine, are also steroid-based. These play an important structural role. They are a major component of membranes that bind cells. Cholesterol plays an important role in the fluidity of the phospholipid bilayer.
They also help absorb vitamins A, D, E, and K.
Some negative effects they can have on health are, because of their insolubility, they tend to be deposited in the walls of the main blood vessels, and this can restrict blood flow. This is associated with high blood pressure. Because they are converted into adipose tissue for storage, a diet too rich in lipids can lead to obesity. These are also linked to other health issues, including diabetes and certain cancers.